Welcome back, everyone, to another deep dive. This time we're taking a close look at two bacterial genera that can be, well, frankly, they can be a bit tricky even for us seasoned microbiologists.
Yeah, I'd agree with that for sure.
We're talking about Aeromonas and Plesiomonas.
Two very interesting groups of bacteria.
And our guide for this deep dive is the Excellent Principles and Practice of Clinical Bacteriology, second edition by Gillespie and Hawkey.
It's a great resource.
It is. I think we'll all agree that these bugs can be tough to pin down sometimes, even with the best resources.
Oh, absolutely. They can really throw you a curveball in the lab.
So to kick things off, let's start with Aeromonas. They're gram-negative rods, which seems straightforward enough.
They're pretty standard stuff.
But then they start to show their wild side, shall we say.
Yeah, they definitely have a few tricks up their sleeve.
They can form pairs and chains and even swarms.
Yeah, they're not afraid to get social.
And their affinity for water. That's a big clue for us, isn't it?
It is. It tells us a lot about where we might encounter them and the types of infections they can cause.
Exactly. It's not just a casual dip in the pool for these guys. They thrive in water.
Right. They're right at home in aquatic environments.
So for us, as medical microbiologists, this means they're not just a lab curiosity, right?
Absolutely.
We're likely to see them in real patients in ways that we need to be prepared for.
Definitely. And that's where the identification challenge comes in.
Right. Even with all of our fancy tools, sometimes nailing down the exact species can be a real headache.
It can be. It often requires a combination of phenotypic and genotypic methods.
Wow.
You know, good old-fashioned lab work combined with the latest molecular techniques.
So experience really counts in those cases.
It does. You need that keen eye to spot the subtle differences.
Okay, so Aeromonas, we've got them pegged as shape-shifting, water-loving bacteria. What about Plesiomonas?
Well, the name might fool you a little.
Yeah, neighbor to Aeromonas, right?
That's what that means. But genetically, Plesiomonas is actually closer to Proteus.
Oh, really?
Yeah, it's kind of a microbial family feud going on there.
So much for being neighbors.
Right. And to make things even more interesting, Plesiomonas had a bit of an identity crisis in the past. It was bounced between the Pseudomonas and Aeromonas genera before finally getting its own classification.
Sounds like taxonomists had a field day with this one.
They did. It just goes to show that science is always evolving and we're constantly learning new things.
But despite its wandering past, Plesiomonas does share some similarities with Aeromonas.
Yes, it does.
It's also a gram-negative rod.
Although it has a bit more of a dramatic flair.
Oh, so?
Well, it tends to form these long filamentous shapes, almost like tiny threads.
So still playing the chameleon card, I see, and still hanging out in those aquatic environments.
That's right. They both seem to have a preference for water.
Our book really emphasizes how this makes both of these bacteria relevant to us as medical microbiologists.
And it makes sense, doesn't it?
It does. We're the ones on the front line seeing patients who might have encountered these bacteria in, say, contaminated water or food.
Exactly.
Right.
And that brings us to the big question.
Which is that?
Why are these bacteria a concern for human health? What makes them pathogenic?
Right. Let's get to the heart of the matter. The danger factor. What makes these bugs tick? And the book really highlights Aeromonas' S layer.
Ah, yes, the S layer.
It's this protein layer that helps them stick to tissues.
Like glue.
Almost. I was thinking more like Velcro.
Yeah.
But yeah, they can really latch on.
Oh, definitely. But the S layer does more than just help them stick. It also helps Aeromonas evade our immune system.
Sneaky.
Very. It can resist complement and even interfere with our macrophages' ability to engulf them.
So they're not just hitching a ride, they're actively fighting back.
That's right. They're not going down without a fight.
And as if that wasn't enough, they also have fimbriae and adhesins.
Right. Those act like grappling hooks, giving them an even tighter grip.
Especially relevant for us because, as the book points out, they can latch onto the gut mucosa.
Not a good sign for the patient.
Not at all.
Yeah.
And then there's the capsule production.
Oh, yes. In certain environments, Aeromonas can actually cloak itself.
Like a microbial invisibility cloak.
Exactly. It makes it even harder for our immune system to detect and target them.
And what kind of environment triggers this cloaking device?
Well, it seems that sugar-rich environments can stimulate capsule production in Aeromonas.
So those sugary drinks might be fueling more than just a sugar rush.
Potentially, yes. It's a reminder that environmental factors can have a big impact on bacterial behavior. Something you, as a medical microbiologist, are well aware of.
We are, but let's not forget the S layer and adhesion factors are just a tip of the iceberg.
Oh, there's much more to the story.
Aeromonas also packs a punch with a variety of toxins.
They're not afraid to use a little chemical warfare.
So spill the tea.
Yeah.
What kind of toxic weaponry are we talking about?
Well, two of the most concerning are cytotoxins and enterotoxins.
Those sound pretty ominous.
They are cytotoxins, as the name suggests, directly damage host cells. And enterotoxins. Those wreak havoc in the gut, causing those classic symptoms of gastroenteritis.
So not only do they stick to your gut, they then start throwing punches.
Exactly. And they're not limited to just those two. They also produce hemolysins, which break down red blood cells, and phospholicases, which disrupt cell membranes.
Okay, now it's getting scary. This sounds like a full-on microbial assault on the body's cells.
It can be, especially in more severe cases.
But here's what I find fascinating. Even seemingly harmless enzymes like amylase can actually act as virulence factors in Aeromonas.
It's true.
Wait, amylase, isn't that involved in digesting starch?
It is, but within the host, it can contribute to tissue damage and inflammation, making the infection worse.
So it's not just the flashy toxins, but also the seemingly innocent players that can turn against us.
It's a reminder that even those everyday enzymes can be hijacked for nefarious purposes.
Makes you realize how complex and adaptable these bacteria are.
Precisely.
And of course, we can't forget about the endotoxin, or LPS, common to all gram-negative bacteria.
Right, that's a key player.
In Aeromonas, it can really ramp up the severity of the infection, triggering a strong immune response.
It can. And then there are the cytophores, those iron-scavenging molecules that help Aeromonas thrive even in iron-deficient environments.
So it's like they're stealing the very nutrients they need to survive from their host.
Quite right. It's a brilliant strategy from their perspective, though not so great for the infected individual.
Okay, so Aeromonas is armed and dangerous. What about Plesiomonas? Does it play by the same rules?
Well, while not as extensively studied as Aeromonas, Plesiomonas also seems to rely on toxins to cause trouble. Particularly enterotoxins. It's all about causing mischief in the gut.
So still a gut disruptor, but maybe with a slightly different approach.
That's right. And it also produces cytolysins, which, as you know, can damage cells and disrupt the delicate balance of the gut microbiome.
And as microbiologists, we know how important that gut balance is for overall health.
Absolutely. It's a complex ecosystem that can be easily thrown off balance.
So we've got these two bacteria, both water-loving, both packing a toxic arsenal, and both with a knack for causing gut trouble. What does this actually look like in patients?
That's a great question, and one that we'll dive into after a quick break.
Stay tuned, folks. We'll be back in a flash to explore the clinical features of Aeromonas and Plesiomonas infections.
All right, so let's talk about what these infections actually look like in patients. What are the clinical features we need to be on the lookout for?
Yeah, let's dive into the nitty-gritty. What are we actually seeing in the clinic?
Well, when it comes to Aeromonas, there are three main species that cause most of the trouble in humans.
Oh, really?
Yeah. There's Aeromonas hydrophila,
Okay.
Aeromonas caviae,
Got it.
and Aeromonas veroni biotype sobria.
The big three. So I guess those are the ones we really need to know.
They are the usual suspects.
Now, we talked about those enterotoxins earlier.
Right.
So I'm guessing gastroenteritis must be a pretty common presentation.
It can be, but there's actually some debate about how often Aeromonas is truly the cause of gastroenteritis.
Really? So it might not always be the guilty party.
That's right. Some studies have found Aeromonas in the stools of both healthy people and those with diarrhea.
So it could just be a bystander.
It's possible. But we also can't ignore the fact that there have been documented outbreaks linked to Aeromonas.
So bit of a gray area then.
It is. And to make things even more complex, the symptoms of Aeromonas gastroenteritis can vary quite a bit.
From
mild watery diarrhea to more severe dysentery with blood and mucus.
And are certain people more susceptible?
It seems travelers, particularly those visiting tropical or subtropical regions, seem to be at higher risk.
So another reason to be cautious about food and water when traveling.
Absolutely. But it's not just about what you eat or drink.
What else?
Well, Aeromonas can also cause a variety of infections beyond the gut.
Oh, right. We were talking about that earlier.
Yeah, they can invade tissues, cause wound infections, even get into the bloodstream.
Okay, let's break those down a bit.
Yeah.
Starting with those extraintestinal infections, what are we talking about specifically?
Well, one common scenario is wound infections following exposure to contaminated water, like swimming in a lake or river, or getting a cut while fishing.
Yeah, I can see how that would happen.
And the bacteria can also enter through existing wounds, like if you have a cut or abrasion and you go wading in a pond or something.
Ouch. That doesn't sound pleasant.
Not at all. And in some cases, these wound infections can progress to more serious conditions like cellulitis or even necrotizing fasciitis.
Okay, now that's scary stuff.
It is. And then there's bacteremia.
Which?
When the bacteria get into the bloodstream.
Oh, boy. That sounds serious.
It can be, especially for certain people
Mm.
with weakened immune systems or underlying health conditions.
And infants.
Yes, infants are also at increased risk.
And how serious can those bloodstream infections get?
Well, in some cases, the mortality rate for Aeromonas bacteremia can be as high as 50%.
50%. That's a sobering statistic.
It is. It highlights the importance of early diagnosis and appropriate treatment.
Absolutely. So you've got wound infections, bacteremia. What else?
Well, Aeromonas can also cause respiratory tract infections, although those are less common. And they've been linked to peritonitis, which is inflammation of the lining of the abdominal cavity.
And HUS.
Yes, hemolytic uremic syndrome.
Wow. So it's not just a gut bug then. It can really affect different parts of the body.
Exactly. It's a versatile pathogen.
All right. So that's Aeromonas. What about Plesiomonas? What are its clinical trademarks?
Well, as we mentioned earlier, Plesiomonas is best known for causing gastroenteritis, especially in tropical and subtropical regions.
Right. We talked about that. So if you're seeing a patient who's recently returned from a trip to the tropics and they're having some tummy troubles,
Plesiomonas should definitely be on your radar.
makes sense.
Mm-hmm.
And is it always from water?
Water is the most common source, but contaminated food, especially seafood, can also be a culprit.
So another reason to be mindful of what you eat when traveling.
Definitely. And while gastroenteritis is its main claim to fame, Plesiomonas has also been linked to other infections, although they're much less common.
But
Well, there have been cases of bacteremia, meningitis, even endocarditis.
those are pretty rare.
They are, thankfully. But it's important to keep in mind that Plesiomonas can occasionally cause these more serious infections, especially in people with compromised immune systems.
So always be on the lookout.
Always be prepared for anything.
All right, we've talked about these bacteria in the environment and in patients. Now let's shift gears and head into the lab. How do we actually diagnose these infections?
That's a great question, and one that we'll tackle after a quick break.
Stay tuned, folks. We'll be right back to explore the world of Aeromonas and Plesiomonas diagnostics. Welcome back, everyone. Now it's time to put your Aeromonas and Plesiomonas knowledge to the test. Are you ready for some exam-style questions?
I hope so. We've covered a lot of ground.
We have, but I think you're all up for the challenge. Let's start with question number 1. What key feature distinguishes Aeromonas from most other Enterobacteriaceae and is crucial for initial lab identification?
That's a good one. Think back to those biochemical tests.
All right, time's up. The answer is the oxidase test Aeromonas is oxidase positive, unlike most other members of the Enterobacteriaceae family.
That's right. It's a simple test, but it can make a big difference in narrowing down your suspects.
Okay, on to question number 2. Name 2 virulence factors that allow Aeromonas to adhere to tissues, potentially leading to some pretty nasty infections.
This one should be a bit easier. Think about those sticky structures we discussed.
All right, pencils down. The answer is Fimbriae and adhesins. These little guys act like grappling hooks, helping Aeromonas latch onto host cells.
And don't forget about the S layer. That also plays a role in adhesion and immune evasion.
Right, the S layer is a key player in Aeromonas pathogenicity. Question number 3. What patient population is most at risk for developing Aeromonas bacteremia, requiring our heightened vigilance as medical microbiologists?
This one's about thinking clinically. Who's most vulnerable to these infections?
The answer is immunocompromised individuals and infants. Their weaker immune systems make them more susceptible to these serious bloodstream infections.
And we know that Aeromonas bacteremia can be life-threatening, so early diagnosis and treatment are crucial in these cases.
Absolutely. Question number 4. Why might commercial identification kits fail to accurately identify Aeromonas to the species level? And what should a savvy microbiologist do in this situation?
This highlights a real-world challenge we often face in the lab. Sometimes those fancy kits just don't cut it.
The answer is commercial kits often rely on a limited number of biochemical tests, which might not be enough to distinguish between closely related Aeromonas species. So what's a microbiologist to do?
Well, you got to go back to basics. Rely on your knowledge of those key biochemical tests and consider using additional methods like molecular techniques for confirmation.
Right. It's all about using the right tools for the job. And finally, question number 5. True or false? Plesiomonas shigelloides infections are always associated with gastrointestinal symptoms.
This one's a bit tricky. Think carefully about the range of infections we discussed.
The answer is false. While gastroenteritis is the most common presentation of Plesiomonas infection, it can also cause extraintestinal infections like bacteremia, meningitis, and endocarditis.
So don't get tunnel vision on the gut. Keep an open mind and consider other possibilities, especially in patients with more unusual presentations.
Great point. So there you have it, 5 questions to test your Aeromonas and Plesiomonas expertise. We hope you enjoyed this deep dive into the world of these fascinating and sometimes frustrating bacteria.
It's been a pleasure sharing our knowledge with you all.
And remember, as medical microbiologists, we play a vital role in diagnosing, treating, and preventing these infections. Keep those minds sharp, those microscopes focused, and stay curious, because there's always something new to discover in the amazing world of microbiology. Until next time.